Okay, hi semua. Now we will do the pre-lab session for the experiment number 2 about the plant tissue. So this will be the learning outcomes in which at the end of this lesson we should be able to identify the different types of the plant tissues, and then we need to compare the structure and also the distribution of the tissues in monocots and dicots plant. Then we need to be able to illustrate the correct scientific drawing for the plant tissue. Okay, and so hopefully that you can try to scan this QR code in which you'll be able to see this particular 3D apps. Okay, so if you scan this particular QR code, firstly you will be able to see the images of monocot and dicot leaves, and then you try to click on the other part about the dicot and monocot stem and also roots. So this will be about the leaves. On the left hand side, it will show you this particular leaf of the monocot plant. Okay, in which if we look at the cross section of the monocot leaf, you'll be able to see these vascular bundles, okay, that are arranged parallel to each other. And on the right hand side, this will be the leaf of the dicot plant, okay, in which we will be able to see the arrangement of the vascular bundle is slightly different. Over here on top with larger structures will be the xylem tissue, and down here will be the phloem tissue. Okay, but usually we will focus more on the stem and the roots. Okay, so hopefully you can click on the menu for the stem and also the roots for the monocot and for the dicot plants in that particular apps just now. So if we look at the cross section of the monocot stem, it will be something like this, in which please take note of the arrangement of the vascular bundles. Okay, the vascular bundles in the monocot stem will be scattered. Yes, scattered. There will be no particular arrangement for the vascular bundles. Okay. Meanwhile, if we look at the cross section of the dicot stem, we will be able to see that the vascular bundles over here will be arranged in a ring, okay, in a circle. Okay, so on the outermost layer will be the epidermal tissue. Okay, epidermal tissue, and somewhere here, okay, will be the ground tissue, which might consist of parenchyma, and over here will be the vascular bundles. Okay, this is one vascular bundle in which we can see down here will be the xylem. Okay, xylem will be having larger spherical structures, larger cylindrical structures. Okay, meanwhile this blue part over here, okay, this one will be the phloem. Okay, and this white section over here is actually referring to the sclerenchyma, the sclerenchyma tissue. Okay, in the middle of the stem of the dicot will be the part that we call as the pith. Okay, the pith, umpulur in Malay. Okay, and then we try to go to the roots, okay, of the monocot and also for the dicots. For the monocot root, okay, if we look at the cross section, we will be able to see that the vascular bundles will be arranged in a ring form, okay, in a circular pattern. So On the outermost layer, you will be able to see the epidermal tissue over here. Okay, and you might also see the root hair. That will be important for absorbing water and also nutrients from the soil. Okay, and over here will be the cortex region, which primarily consists of the parenchyma cells, and then you will be able to see the vascular bundles. Okay, over here, in which these blue structures will be the phloem, and towards the middle will be the xylem, somewhere here, and the middle, and in the center region will be the pith, and the umpulo. Again, it consists of the parenchyma tissue. Okay. Meanwhile, if we look at the dicot root, okay, on the outermost layer, still it will be covered by the epidermal tissue. Okay, and we can also see the root hair. Okay, here and there, for the absorption of water and nutrients from the soil. And over here will be the cortex region, consisting of parenchyma cells. And towards the middle, you will be able to see the vascular bundles, in which we will be able to see something very unique about the dicot root, in which the xylem tissue will produce star-like shape. Okay, seperti bintang, star-like shape in the middle of the dicot root. Okay, this yellow colored structure, this will be the xylem tissue, and there will be the phloem tissue as well, the blue colored ones somewhere here. Okay, so this will be the cross section of the monocot and also the dicot root. So if you look through the app that is given through the link just now, you'll be able to see the three-dimensional image for these structures. Okay, and so let's look at the introduction part. Okay, in which you need to try to answer some of the questions given. Okay, so for the introduction part, question number 1, you need to state 3 main characteristics of the plant cell. Okay, so you should notice that over here will be the plant cell wall. Okay, so every plant cell should have the cell wall lah. Okay, then over here will be special organelles. Will be the special organelle that can only be found in the plant cell. That will be the chloroplast. So we say that plant cell should be having the chloroplast. Okay, in general, And another one will be this structure over here, in which we can identify this as the large permanent central vacuole, only in the plant cell. Okay, now let's move on to the second question. Okay, the plant tissues are divided into 2 groups, which are meristem and the permanent tissue. Okay, so what will be the main difference between the meristem tissue and the permanent tissue? Okay, basically the meristematic tissue or the meristem tissue undergoes cell division, okay, mitosis, in order to produce new cells. Okay, meanwhile, the permanent tissue consists of matured and differentiated cells that will be having specialized structure and function. Okay, so this will be the main difference between these 2 types of tissues in the plants. Okay, part 3, question number 3. So if we look at this figure number 1, okay, we should try to identify the type of tissues. Okay, for the plants, basically the meristem tissue and the permanent tissue together with the vascular tissue. Okay, so over here will be the apical meristem. The same goes for the tip of the roots. Okay, the tip of the roots. So at the end of the roots and also at the tip of the shoots, okay, hujung pucuk, hujung akar, there will be many meristematic cells that will be undergoing the process of cell division. Okay, so apical meristem will be at the ends of the shoots or at the end of the roots. Okay, now if we look at this stem, okay, this stem area, if we do a cross section, we will be seeing something like this. So what will be these tissues over here? Okay, the outermost layer should always be the epidermal tissue. Yang paling luar, the epidermal tissue. And filling up, okay, the internal parts of the plants will be the ground tissue. Meanwhile, the dark ones over here will be the vascular tissue consisting of the xylem and also phloem. Okay, and if we look at the cross section of the root, okay, again, the outermost layer will always be the epidermal tissue, and filling up this part of the plant will be the ground tissue, and in the middle usually will be the vascular tissue, the xylem and the phloem. Okay, now let's move on to question number 4. Okay, if we refer to figure 2, okay, the structure number 1 and also structure number 2 will be part of the vascular bundle. Okay, normally if we refer to the vascular bundle, the lower part will be having larger vessels, okay, and that will be the xylem tissue. Meanwhile, on the upper part of the vascular bundle will be having smaller vessels, and that will normally referring to the phloem tissue. Okay, so number 1, structure number 1 will be the xylem tissue, which we can find in the stem, the root, and also the leaf of the plant. Well, structure 2 will be the phloem, and we can also find this phloem tissue in the stem, the root, and also the leaf of the plant. Okay, over here will be the cortex region. Okay, the cortex region. Meanwhile, in the middle will be the pith. Okay, so the cells that will be found over these two areas will be the parenchyma cells, which will form the parenchyma tissue. Okay, over here at this cortex region and also at the pith, and the parenchyma tissue, which can also be found not only in the stem but also in the roots and also the leaf. Okay, and just below the epidermal layer, okay, the outermost layer will be the epidermal tissue. Okay, so just below the epidermal layer, you will find this tissue number 4, which is actually the collenchyma tissue. Okay, normally it will be at the stem area. Okay, meanwhile, somewhere at the vascular bundle will be another type of tissue that will be the sclerenchyma. Okay, and we can see the difference from this particular diagram. Okay, between the parenchyma, the collenchyma, and sclerenchyma. Basically, you just try to focus on the cell wall, the cell wall of these tissues, in which the parenchyma will be having thin cell wall, while the collenchyma has got thicker cell wall, but the thickening is uneven. and the thickening is uneven. Meanwhile, for the sclerenchyma, there will be this thick cell wall, which normally referring to the thick secondary wall. Okay, so that will be the difference lah. If we look at the condition of the cell wall of these three types of plant tissues. Okay, let's move on to question number 5. Okay, what will be the main tissues of the vascular bundle in the plants? So this is very straightforward. The xylem and the phloem. Okay, so as I have told you just now, the xylem will be showing larger vessels. Okay. Meanwhile, the phloem will be smaller. Okay. And now let's go to the experiment part of your pre-lab module, in which you will find these 4 diagrams. Okay, try to identify whether this will be the roots or the stem, whether this will be the diagram for the monocot plant or the dicot plant. Okay, so for diagram 1A, okay, this should be the dicot stem. Okay, the dicot stem. Meanwhile, for diagram 1B, this will be referring to the monocot stem. So basically, how can we differentiate between these two? Okay, it will be the vascular bundles. As you can see over here, the vascular bundles for the dicot stem will be arranged in a ring or a circular pattern. Okay, so on the outermost layer will be the epidermal tissue or the epidermal layer. Okay, and over here will be the pith, which consists of parenchyma cells, and somewhere here will be the cortex, also consisting of parenchyma cells, and over here will be the vascular bundles, which consists of the xylem over here and the phloem somewhere here. Okay, the dark ones near the vascular bundles, the dark ones over here, that actually referring to the sclerenchyma tissue. Okay. Okay. Meanwhile, the monocot stem will be different because the vascular bundles are scattered. Dia berterabur within this cross section of the monocot stem. So again, on the outermost layer will be the epidermal tissue or the epidermal layer. Okay, and all these small dots will be actually referring to the vascular bundles which are scattered lah, tersebar di sana sini. Okay. Meanwhile, what about 1C and 1D? So these two will be the roots actually, and the roots. So which one is the root of the dicot? Which one is the root for the monocot? Okay, we should try to take note of this structure in the middle over here. Okay, as I have mentioned just now, if you see a star-shaped structure in the middle of the root, then that must be the root of the dicot plant. Okay, this will be the xylem. The xylem that is arranged in a star-like pattern, seperti bintang in the middle. And over here, also near the xylem, will be the phloem. This blue colored area over here between the xylem tissue. Okay, so on the outermost part will be the epidermal tissue, and here will be the cortex region. Okay, consisting of parenchyma cells. Okay. Meanwhile, this diagram 1D will be showing the monocot root, in which the vascular bundles, the xylem and the phloem, will be arranged in a ring or in a circular pattern. Okay. So on the outermost part, again, it will be the epidermal tissue or the epidermal layer. Inside will be the cortex region consisting of parenchyma cells. In the middle will be the pith of the monocot root, also consisting of parenchyma cells. Okay. Meanwhile, the vascular bundles will be consisting of xylem and phloem. So the xylem will be the ones that are larger over here, the larger circles. Okay. Meanwhile, the phloem will be in between, with smaller vessels. Okay. So hopefully you can identify and can differentiate between the stem of the dicot plant and the stem of the monocot plant, and also the root for the dicot plant and the root for the monocot plant. Okay, now let's move on to question number 2, okay, in which we need to identify and label the different type of ground tissues. So remember, when we talk about the ground tissues, it will be about the parenchyma tissues, the collenchyma tissues, and also the sclerenchyma tissue. Okay, so over here will be the collenchyma. Okay, remember that on the outermost layer it will be the epidermal tissue, and just below the epidermal tissue will be the collenchyma tissue. And filling up this part of the plants will be the parenchyma tissue. Meanwhile, somewhere near the vascular bundles over here, and also over here will be the sclerenchyma tissue. Okay, and just to show to you that these larger circles will be the xylem. Xylem. This one. Okay, this one xylem. This one is also the xylem, this one, and so on. The phloem will be smaller. Okay, this will be the phloem area, this one, and also this one over here. Okay. Okay, and then for this particular diagram, again the collenchyma will be just below the epidermal layer. Okay. And then over here will be the cortex region consisting of parenchyma cells or parenchyma tissue, and again this will be the sclerenchyma, okay, near the vascular bundles. Okay, and what about this these diagrams? The cortex of this particular root, this will be the root of the dicot plant. Okay, why? Because we can see that the xylem will be showing a star-like shape, seperti bintang. Anyway, this cortex region over here consists of parenchyma cells, so this will be the parenchyma tissue. The same goes for this root of the monocot plant. Okay, the cortex region will be actually referring to the parenchyma tissue. Okay, move on to question number 3. Okay, differentiate the arrangement of the following structures. Okay, the arrangement of the vascular bundles in the monocot and also in the dicot stem. Okay, as I have told you just now, the vascular bundles in the monocot stem will be scattered. Meanwhile, the vascular bundles in the dicot stem will be arranged in a ring or in a circular pattern. Okay, what about the xylem and the phloem in the monocot and the dicot roots? In the monocot root, the xylem and phloem will be arranged in a ring pattern or in a ring form. Meanwhile, the xylem will be arranged like a star shape in the dicot root. Okay. Now let's move on to the question number 4, in which we need to differentiate between the parenchyma tissue and the collenchyma tissue, and also the sclerenchyma tissue. So the first part will be about the presence of nucleus. Okay, we need to understand that parenchyma cells will be living cells, and so the nucleus will be present. The same goes for the collenchyma tissue, in which the collenchyma cells will also be a type of living cells. So nucleus is also present in the collenchyma tissue. However, for the sclerenchyma, we need to understand that the sclerenchyma cells are actually dead cells when they become matured. Okay, and so nucleus will be absent because they are dead cells when they reach maturity. Okay, the next part will be about the shape of the cell. For the parenchyma, it is isodiametric. Well, for the collenchyma, the cells will be having this polygonal shape, and from the side, the cell will be elongated. Seems to be elongated with tapered ends. Okay, memanjang dengan hujung yang runcing. Tapered ends means hujung yang runcing. Well, for the sclerenchyma, it is also elongated with polygonal shape and tapered ends. Okay, this is for the fiber cells, because we know that sclerenchyma will be having two types. Okay, the first type will be the fibers, and the second type will be the sclerites. So for the sclerites, the shape will be isodiametric, or they will be having irregular shape. Okay, let's move on to the third structure, which is about the cell wall. Okay, this is very important, and you need to take note that for the parenchyma, there will only be distinct and flexible primary wall. Okay, there will be no secondary wall for the parenchyma, only thin primary cell wall, and the cell wall consists of these substances: cellulose, hemicellulose, and also pectin. For the collenchyma, the cell wall is also a type of primary cell wall. Okay, there will be no secondary wall for the collenchyma. However, the difference between the collenchyma and the parenchyma is that the cell walls will be thickened. Okay, but the thickening is uneven. Okay, tebal tapi tidak sekata. Normally the thickening will be at the corners, dekat perpenjuru sel. At the corners of the cell, there will be this thickening. Okay, due to the deposit of cellulose, hemicellulose, and also pectin. Okay, and for the sclerenchyma, it will be very different because there will be a secondary wall. So sclerenchyma will be having a primary wall and also a very thick secondary wall, and the secondary wall is normally thickened by lignin. So we can say that the secondary wall is lignified. Okay, lignified because there is a substance known as lignin. Okay, that causes the cell wall to be very thick. And another part will be about the arrangement of the cell. For the parenchyma tissue, the cells are loosely packed. This means that they have large intercellular air spaces. Okay, ada ruang-ruang udara di antara sel-sel parenchyma. But for the collenchyma, the cells are closely packed, and so there will be less intercellular airspace. And for the sclerenchyma, the cells are tightly packed together and there will be no intercellular space at all between the cells. Okay, and so this will be the differences between the three important tissues of the plants, the three important ground tissues. Okay, and so that's all for this pre-lab session for experiment number 2. Okay, we will do this experiment 2 virtually next week, inshallah. Okay, that's all for now. Thank you.